I am Dr. Roberto Ruano. I am the assistant professor at UConn and also assistant director of the Connecticut Convergence Institute for Translation in Regenerative Engineering. Today is the third webinar in our series, Fundamentals for Clinical Trials of Tissue Engineered Medical Products. I am extremely pleased and grateful as I acknowledge our two speakers today that will be doing the honors. They will be presenting the topic of clinical trial and design. These are my colleagues, Dr. Grady and Dr. Wu. The title of the talk today is Design and Statistical Rigor in Clinical Trials for Regenerative Medicine. Dr. Grady is a senior professor and statistician with more than 25 years of experience as lead biostatistician for many NIH-funded programs ranging from basic science to program grants and population-based interventional trials. He currently directs the Biostatistics Center for the Connecticut Convergence Institute for Translation in Regenerative Engineering. Dr. Wu has been conducting research in clinical populations, screening, recruitment of thousands of participants, including patients with chronic diseases. Her current research involves clinical interventional studies, including development of strategies to improve retention of patients with substance abuse disorders. And recently, she has been co-principal investigator of programs on the reduction of cardiometabolic risk, particularly in populations of African and Hispanic American descent that have risk factors for type 2 diabetes. So in keeping with our goal, these presentations are part of the involvement of UConn as a Tier 1 member of the Army. It has been done in partnership with Army to reach the membership. I would ask the audience to write comments or questions in the chat tool of the platform. I will be reading those questions at the top of the hour. We are going to have first Dr. Wu do her remarks, followed by Dr. Grady. And then finally, this event is being recorded and archived at the website of the Connecticut Convergence Institute. And also, it will be available to the members of Army as well. So thank you for your attendance, and I will pass this on now to Dr. Wu.
Okay, I'm going to take my mask off, and thank you for inviting me to this talk. So I'm going to pull up my slides. Can you see it? Yeah.
Yep, we can see it.
Okay. So, and thanks for Dr. Riwano's introduction. And I have been doing human subject research for 20, 30 years. So the first part of the talk, we're going to introduce the major component, basic component of interventional trials. And so part two, Dr. Grady is going to talk about how to incorporating statistics in research protocol. So for the part one, the learning objective is first we're going to introduce what interventional studies are, and we want to understand what the phases of interventional study or clinical trials. And we also will talk about the major component of clinical trials. You already heard probably about randomization, blinding, power analysis, all these the terms. So we'll hopefully have some definition for you, and then you have a better understanding of that. So you can see the slides. I have both interventional studies and clinical trials here. it's confusing. And I think in the field, some people like to use the term interventional studies because it is a study, and interventional here is more clear. It's intervention, and clinical trial doesn't give you what that means. It is in the clinical field. So throughout this the talk, I probably will interchangeably using both terms. And plus, for interventional study, it could apply to animals too. And what probably a lot of you doing research in this way, that the clinical trial are also more specific to humans when you do an interventional study. So I want to ask a couple questions here, and I know you are not allowed to talk, but you can use your chat box to say, did you ever conduct human studies? Let me see if I can see the chat box. Is there any way I can actually see the chat box? Does anybody say yes or no? I can't see the chat box. Okay. And just to think about it, I would like to know a little bit more. And have you ever conducted or participated in any interventional trials in human? And oh yeah, here is the chat. Nobody answered the question. That's okay. So I will keep talking. So what are interventional studies? How do we define it? And by definition, well, it's implied experimental study, right? You do studies on animal, you give it an agent. So interventional study on humans, that's what intervention about. But the goal is the goal of the trials, interventional studies, is to determine the efficacy or effectiveness of an intervention. This continuum of disease. So from disease-free people, you want to see if that any intervention can interfere the development progress to a disease. And also, if you study on sick people, and you want to see if there's a reduction of the prognosis, like the severity of diseases. For example, in regenerative medicine, we have trying to come up with agents can reduce severity of osteoarthritis in knee, in hip, or in anywhere, because we don't have any intervention, pharmacological intervention available yet. So it's a human subject study, so we need a cohort of individuals instead of animals. So how do we identify the cohort of individuals? So some study needs the disease-free people, but usually at a high risk. Thinking about if a completely healthy person, if you want to use an intervention, you don't know if this person will develop disease or not. So that's why depends upon what you really study. So you want to look at select a cohort of individuals, even though it's disease-free, but potentially high risk. And also the other depends what your agent is, what your study is. So you want to probably select some people with particular disease. And what you do, you intervene with using a suspected prophylactic in disease-free people, so they can intervene the disease process, or a treatment. And I think both apply to regenerative medicine too. It's too bad I don't know what you do, what kind of research you do in actual regenerative medicine. That'll be really if in person I can ask you questions, so I will know what kind of agents you study, what kind of population will be better for your what you are doing. And then, what is the goal for interventional trial? Really, the ultimate goal is trying to establish the causality, right? You have the agent, you interfere the process, but usually it's very expensive because it, for interventional study is an experimental study. It's implied, it's a prospective study, so you have to follow people over time, and it's time consuming with recruiting patients, and the patients has to stay in the study, and also you have to address very narrow questions, and potentially when we translate from laboratory study to human, it can have some harm from the intervention. So that's a key. Safety is a key. We want to make sure any agent is studied applied to human. That's the one thing that's the priority. And for any intervention trials should only preserved for relatively mature research questions. And if you haven't done small animal study, if you haven't done big animal study, it's probably not ready for human study yet, even though it's a fantastic idea. And because we need to prove to make sure and the intervention will benefit the individual and the society. So the big scheme of the interventional trial, we probably know we started with cohort of people, so the population of people. And then you select a group of people with or without disease, probably at high risk of that disease. And we give a treatment, we give a placebo, and probably most people has this concept about what intervention study is. And, in terms of reducing biases, we randomize this treatment to the people are randomized to the treatment and placebo group. So you compare the outcome down the road in the future and see if those individuals, the disease being cured or not cured, or still have the disease. So what we are hoping to get. So we're going to do a little bit detail about what are the key components here. And the next topic I'm going to talk about is what are the phases of clinical trials. You probably all heard about the phase 1, phase 2, phase 3, phase 4, sometimes they call phase 0. And so this is just give you a little overview of the stages of testing on immunotherapy. So first is the preclinical. Probably a lot of you engaged in this type of research is a biological investigation in cells or animals. So when we transfer that into the phase 1 study, phase 1 is usually unblinded, uncontrolled study in few volunteers. A lot of times, usually it's in just a few really healthy people. A lot of times, 10 or 15 healthy men. And sorry, it's my daughter. She's studying at home, so maybe she has something urgent wants to talk to me, but she's okay. Where I was, phase 1 study. And the phase 2 is a small, randomized, controlled, blinded trial to test tolerability and different doses of the intervention. This phase of a study usually takes about it's still a small number, maybe 30, 40 people, depends upon, up to 80 people, depends upon what you study and what's the dose difference you want to detect. And phase 3. So now we all heard about, all this vaccine study for COVID-19. And those are all the phase 3 study. And, for the one from Pfizer, we know each arm has 5,000 people in it. And so they have hundreds of thousands of people in this study. And so it's a large randomized controlled blind trials to test the effect of therapy on clinical outcome. So, and the phase 4 is a postmark surveillance study. So a lot of times they do this type of study using pharmacy data, using health insurance data, and sometimes the pharmaceutical company combine all this data to look at the effectiveness of the clinical trials, and also other side effects for particular pharmacological agents. So if you have questions, and we can answer that later. And so after we introduce all the different phases of a clinical trial, so how do we conduct clinical trials? What are the major components we're going to look at here? How do we understand it? I always emphasize on population. So how do we select the population? And at baseline, how do we select the measures? And what are the end outcome? Probably a lot of times you know what the end outcome you want to do. And during this study, how do you eliminate the biases? So how do we conduct randomization and blinding? And when we look at an effect of the trial, they can have other known effects to impact the magnitude of your study results. And there are other issues here. Power analysis will be given by Dr. Grady in the part 2. So I'm going to make another diagram and saying we have a target population, just like I'm a visual person, so it's easier for me to have a graph to show you what I'm thinking about. We're talking about the selection of people, so we have a group of people, and with the actual subject, this whole process, there's a sampling biases too. And so we want to have the good entry criteria. For animal study, it's easy, relatively, we know we have whatever the mouse model, animal model you want to study, and those are especially breed with very particular characteristics. Humans, everybody has so many different characteristics. Every individual is different from others. So you try to select a more homogeneous group of people. So the entry criteria is really important to consider when you study the human subject research. Then, you have the exposure or the treatment. So how do you eliminate this biases? So in the previous slides, we talked about randomization. And so then we have the outcome. So we have to select the outcome, and from the exposure to outcome, we have to eliminate the treatment biases. So the strategy we use is blinding. So next few slides, I will explain the detail about what are the things you need to consider for each aspect of when you conduct the clinical trials. So for entry criteria, so subject should be high risk. I've been repeating this. And like for example, if you want to study some kind of agent to reduce knee of osteoarthritis, you probably want to select the people who have arthritis or who prone to have arthritis. There are some characteristics, right? So those people are more likely to benefit from your intervention. And the other thing is we have to really consider those people are not be harmed by the intervention. And the other key is if you recruit people, they are very high risk and they are more likely to drop out. People, if you have a 1-year study and some people, especially young people, are more likely to be mobile because of the job, because of other things, getting married, move to out of the area. So a lot of times we want to ask people, are you planning to stay in the area for a year? That's the duration of the study. And if they don't, that can be a exclusion criteria. They cannot be in the study. Because unless your study is remote follow-up is okay, if they have to come in to do checkup, to do getting biologic samples from you at each interview, then most people you will have a higher rate of dropout. And so they are likely to follow up to the full trials. So a lot of the trials, they anyway. So more specifically, and our goal choosing the entry criteria is you are really trying to maximize in the feasibility and at the same time the generalizability. Sometimes this is a balance. And the important difference between observational study versus the clinical trial is first we first do no harm, and if any, so that's why IRB is really important to go through the risk and the benefit. I think the first lecture is about the IRB, so Myra probably talk a lot about how do you balance risk and the benefits in human subject study. So that's what we said, the safety of intervention or of withholding intervention must be carefully assessed for inclusion and exclusion criteria. And then choose the target population for which outcomes is common. And for example, if you choose a study of ADSC intervention trial, you probably should choose the people who are more likely to benefit with more severe knee arthritis. And when you're doing a study, if you don't get a good baseline measure, and you don't know where your patient or your study subject is or are, it's very hard to compare, right? So the measuring the baseline information, collecting the tracking information is really important. And for experimental study, even for our field intervention study, tracking information, what degree of the tracking information you can get for this patient or for this participant is really important. And in the past, when I did the substance use information, we're actually asking about their church attendance, where they hang out, because the substance users may not have a home, and we need to know their social setting. We usually getting like a 2 or 3 phone numbers, and sometimes they have the consent about we can search for online to get their information. So there are so many different ways to get patient information. But if you study a clinical population, and if this patient's coming back, if your patient's coming back all the time, and when we did the clinical study, we tried to align our interview with their clinical time too, and clinical appointments. So a lot of different ways you can keep tracking your patients. But if you're really doing any study, you're working with some recruiters or some companies or people with experience, they can give you a lot of tips about how to do that. So we need to know what kind of patients you are recruiting or subject you are recruiting, so their demographics are very important. And variables affect the risk. So for example, if you study some agent in an arthritic population, probably some other pain-related symptoms or conditions could affect that risk too. So you may have to consider very long list of the comorbid conditions. And so the other thing is because this is a clinical trial, besides questionnaires, and you probably want to also collect some biological samples. And the measure of outcome. So the measure of outcome at the start of the study. So you don't want, because you want to know, for example, severity of the outcome. Severity of arthritis is reduction of severity of arthritis is your goal, one of your goal outcome. So at the baseline, you must have it measured. Then you can, at the end of the study, you can compare, right? Ooh, I'm only 30 minutes into my talk. So the other thing is, whatever the question you're asking needs to be parsimonious. So you don't want to introduce extra survey or questionnaire burden to your patients, because they come to the study no matter how much you reimburse them, they are volunteers. So their time, the burden, you need to respect. And so traditionally, what are the outcome measures? The ultimate outcome is mortality, and some other outcome is morbidity, what kind of disease they have. And some other things you study, probably speed of the recovery, hospital stay, length of a hospital stay, length of ICU, length of stay in ICU, and also quality of life, and also patient satisfactory, especially for regenerative medicine. Sometimes we cannot, at this point, we cannot get direct biological measure yet. For example, and you may want the quality of life, for example, pain, and the satisfaction of life, improvement of functional perception of their functionality, and also objective measure. They are all very important outcome for us to measure. So in terms of regenerative medicine, I'm thinking about pain reduction, and all this you need to measure at the beginning of the research, throughout the research, and the end of your clinical trials. Depends on what you really choose to measure. Pain reduction is one of the things if you study osteoarthritis, and the reduction of the severity of a disease, and the improve functional status, and more biologically, growth of tissue, growth of bones, and you probably have more ideas than I have what else we can measure here. So what is randomization? I think everybody already know just by looking at the number. The key is randomization is a key to the successful intervention. So control for the known and the unknown confounders, and remarkably how powerful this is. And there are so many different ways to do the randomization, block randomization and stratified randomization. When you're actually doing a study, so probably you should always working with a biostatistician. This is just very simple list, but randomization can be simple randomization, can be a very complex randomization technique you use. Binding. So what's a binding? Binding means the researcher or the subject don't know what they're getting. So this is as important as the randomization. So it protects the trial from treatment difference other than the randomized treatment, and also the bias assessment of the outcome, because we don't know what you are getting. And in the co-interventions, so the knowledge of assignment can influence other behaviors. So we'll talk about later. So the biased assessment of outcome, it's not a problem. Very hard outcome such as death, you can get it. And so in terms of binding, if you do a medication study, and even it's the study I mentioned earlier on any regenerative medicine, if you have any pills, any device, if you injection of like a stem cell, type of stem cell to the people, so the identical placebo like injection should be similar to what you're going to inject to the patient. So that's a placebo, tablets or injection or fluids. And you do need a system to break in code fast in case the adverse reaction. Usually this is in clinical trials, this is one of the must you have to design. And you need a system to report side effects, and this is all IRB required. So if you're really truly doing a study, so statisticians and regulatory people can help you to make sure this is in place. And when study is over, and the subject to guess what they got, and if too many are correct, then you must discuss maybe the blinding is broken. So this is a big scheme. At the beginning, we're talking about the different things. So this is really a flow chart of when you do a actual clinical trial. You selected the population, you created eligible criteria, you recruit patients, but every step is very time consuming. So hopefully you did everything right, and you have a good outcome, bad outcome, and your intervention actually can be effective. So, however, the effect you get from the clinical trial may not be the true effect you are seeing. So why is that? Because so it can be patient. Some people have a certain disease can naturally get better, right? And can be a Hawthorne effect. This is actually in 1956, and one of the researchers looking at the Hawthorne electrical factory, looking at people being observed, actually improved. Once the intervention goes away, the effect goes away. So the intervention doesn't have any effect. It's really because people being observed, they are improving their behavior, so that the outcome improved. And so placebo effect, everybody heard about that. I'm not going to elaborate. Then the last one you see is this portion could be the treatment effect. So if we have all this improvement and we see only top part is really the treatment effect, what can we understand? What can we do to tease out all this Hawthorne effect placebo effect. So for history, so you have sick people and as your control group. So you can monitor the same people with your same sick people in the study and if they get better or not. Compared to the, and with Hawthorne effect, if you're also enrolling the sick people and observing, don't give them any medication and see if they change their behavior or not, because they're in the study. And for placebo, and we know we give a sugar pill. It's not really sugar, but the pill looks like what you're giving to people, or injection, the fluids looks like it, like for you. And then the last effect is the rest is the efficacy study. I'm going to end here. I know I went through this quickly, and Dr. Grady is going to give the second part of the talk about how, we can discuss this. Yeah.
Good afternoon. Hope you're enjoying your lunch. So I'm going to talk a little bit about the role of statistics and working with statisticians and when to involve them and that kind of thing here. So here's my talk about creating a research team, whether or not you want to put a statistician on your team. Hopefully you'll want to at the end of this talk. A little bit, and then how to prepare your protocol relative to biostatistics. So you're planning your study, you're creating your research team, you're the PI, you've collected some collaborators, some substantive experts, and then you got to decide whether you're going to include a statistician. And I would say that it's a good idea to include a statistician, especially if you're going to be presenting grants to the FDA, because they're going to have statisticians on their end looking at things, and it's going to really help if you've got a card-carrying statistician on your team. Try to involve them early in the process, because they can help you design your study and help you to focus aims and hypotheses, and I'll give you some examples of that along the way. You're going to need sample size justification and a statistical analysis plan for any of your IRB protocols. So that's just another area where a statistician will be able to help you. I'm going to talk about the flow of information going from specific aims to statistical analysis, and where statistics plays a role, and we'll be talking about primary outcomes, which are often designated with a Y, and explanatory variables or interventions, which are typically designated by an X. So let's talk a little bit about aims and hypotheses. So when you're planning your study, you're going to be formulating your specific aims. Again, try to include a statistician early in this process while the aims are hypotheses are being created. It's not so great to bring them in afterwards, because then they're really not helping you as much. Can save you time in the sense that if you have to recraft aims or hypotheses to make them testable, or the statistician weighs in later on, to change your study design, that can be a little bit of a waste of your time. As this arrow indicates, this is not always the case. Sometimes I'm brought in a little bit too late to be helpful, and that's too bad for the study and for the investigator team. In terms of focusing hypotheses, hypotheses need to be worded in a way that a statistical test can be applied. So sometimes I work with people and we have to craft that so that it can be worded so that a statistical test can be identified for that particular aim. I'll give you an example here. This is a basic scientist I've been working with for a long time. He studies a lot of biological pathways for polyp development and cancer, and he's a basic scientist, but he sort of started getting into clinical work. So we were talking about a grant once, and so I was trying to figure out what his aims were, and some of this was done through email and some of it through in person. And he said, well, I want to determine the status of polyps in the distal and proximal area of the GI tract among colorectal cancer patients. So immediately, I don't know what determine the status is. That's not something you can apply a statistical test to, right? So I come back and I go, well, what do you mean by that? And he says, well, I want to compare the distal versus the proximal regions. So I go, okay, so you want to compare the mean number of polyps in the distal versus the proximal regions of the GI tract. And then he goes, yep. And I go, is that in the same person? He goes, yes. So I like to think of data sets and what my data set's going to look like. So this is what the data set would look like. Each subject with a measure of polyps in each of those regions of the colon. So then I follow this up and I go, do you have any groups in this study? Is this just a single group study, or do you have any groups? And it comes back and, well, of course I have groups. I have to compare these subjects between stage 1 and stage 2 cancer. So then I might say, so focusing this a little more, do you want to compare the extent to which the distal versus the proximal regions are like or different between the stages? And he says, yeah, that's it. That's what we want to do. So now, if you look over here, we've got, now the data set has two groups. It has a stage, another variable in it, and it's got stage 1 versus stage 2. And so that would lead us to a hypothesis like this. Subjects with stage 2 cancer will, on average, have more distal polyps than proximal polyps, compared to stage 1 patients who will have an even distribution of polyps in their distal and proximal regions. So now I'm thinking of a data set that might look like this. And when I give this talk to students, I ask them, well, after focusing that hypothesis, it sounds kind of complicated, but in the end, we can create these difference scores on the far right, and so this can just boil down to a 2-sample t-test on difference scores. So it looked pretty complicated, but in the end, maybe we can apply a somewhat standard and simple statistical test, which is what the agencies like the FDA love. So hypotheses, again, in most cases should be worded so that they're testable with a statistical test. It's good to show which group is going to do better than the other, directionality, and maybe how much better in terms of a percent. So you might say group A response will be 25% larger than group B, or that would be like a numerical example, or you might say that group A will exceed some threshold more often than group B. That would be more like a categorical or binary outcome. So now you've got your hypotheses and your aims down, and you got to start thinking about preliminary data. Do you have preliminary data? Does anybody in your lab have preliminary data? So here we want to know whether we have preliminary data and results. And these can be critical for showing your proof of concept. And the front end of the grant, when you're trying to show background and significance, and then the statisticians really need it for sample size calculations. If you don't have any, sometimes you have to go to the literature. And I'll have an example of that at some point, about how we did that. So if you've got preliminary data, your aims and hypotheses, you feel comfortable with those, now it's time to talk a little bit about your sample size and power. So what is power? A very quick review. So every time you plan an experiment and you start thinking about how you're going to set a p-value to be significant or not, and you start thinking about, I want to power this to a certain level, what you're really doing are you're thinking about your type 1 and your type 2 error rates. So your type 1 error rate typically tends to be about 0.05. That's the error rate that most people are willing to take for a type 1 error. And most people are willing to take about a 0.2 for their beta. That's the rate that they're willing to take for a type 2 error. And then power is defined as 1 minus beta. So that's where you get this alpha equals 0.05 and 80% power terminology from those two, by setting those two error rates at 0.05 and 0.2. So that's just a quick background on how power is defined, but it really gets back to the fundamental type 1 and type 2 error rates that you're willing to make. Now sometimes you want to make sure you don't miss anything, so you may want to overpower your study. You may want to set your power to 0.2. You don't want to be saying there's no difference. You want to make sure you're ruling out no difference. If you're looking at multiple outcomes in a study, you may want to set your alpha a little higher. So there's reasons why these are not always the industry standard 0.05 and 80% power. Why do we have to do a power analysis? Is it really necessary? For smaller studies, it might not be. For pilot studies, if you're just going to pick 10 people per group, there's really not much you can do with that in terms of statistical tests and power. But it does give, for anything bigger than a pilot study, like phase 1 studies, phase 2 for sure, it gives IRB and grant reviewers, FDA reviewers, an idea of how likely you will be to find significance, which again relates back to your type 1 error rate, or declare no difference at the end of the study. And of course that relates to your type 2 error rate. Think about if your sample's too big, you might be declaring things that are significant that really aren't. And if they're too small, you might be missing things. You might be saying there's no difference when really you had no shot at ever declaring those differences because you were underpowered. And in terms of IRBs, the obvious reason for a sample size calculation is that you're justifying the number of individuals exposed to risks in your study, and that would also apply to animals for the animal IRBs. How many animals you're sacrificing for the study and if that's justified. We talk about effect sizes when we do power analysis, and that could be the difference in mean survival. That could be difference in means of a drop in systolic blood pressures, proportions healed. It could be odds ratios. These differences need to be both clinically relevant but also realistic. So at the end of the study, if you're showing a difference, you want that difference to mean something to the clinical people and not just a p-value that it meets. But you don't want to be overly optimistic, because if you say that you're going to have a really big difference in your study and then somebody does a power calculation for that really big difference, and it comes back that your difference is smaller than that, you'll have an underpowered study. So there's a little give and take with this. So what's the role of the investigator in sample size calculations? Here's an example for categorical data. If you're looking at the proportion healed, a standard treatment, which may be healing half the population or half of your patients, your new treatment you think is going to be 75%. Those are numbers that you can't expect the statistician to come up with. These numbers are numbers that need to be supplied by the investigator. the last thing you want is your statistician coming up with these numbers. And I've been asked to do that, by the way. Same thing for continuous data. Let's say you're looking at mean change from baseline of something. Here's maybe a BMI improvement for a new diet versus a standard diet. And we need to figure out some sample size calculations. What we're going to need to look at what we think the difference is going to be. Here the difference would be 2. We also need standard deviations, and that's where the preliminary data comes into play. So your statistician will be saying, well, I can't do sample size calculations without standard deviations and some reasonable deltas, and that's where you both have to work together. Here are some practical issues. What if you only have access to a certain number of subjects? What if you only have 1 year to complete a study? You're under a pressure, a deadline to get something through. Sometimes your N can be limited. Well, you can sort of work backwards with what you have available in terms of resources and see whether you think you can detect that difference. So here, let's look at an example. So let's say you have a certain amount of resources and you think you can have about 20 to 30 subjects per group. And let's say you have a categorical outcome for some response to a treatment or an intervention. So that's like a yes-no type of question. And you say, well, how big of a difference would I have to see in order to get significant result between 2 groups, let's say with 20 per group? So you create a table. So this is what it would have to. And if you start playing around with the numbers, if you have 20 per group, this is how big that treatment effect would have to be. It would have to be 15 yeses that were cured under treatment compared to 5 no's, and then that would have to reverse itself. And that is a huge odds ratio. That is an odds ratio of 9. You probably don't need to do a study if your drug is that effective. So, but that was the first arrangement of those numbers giving 20 per group that gave 84% power. So another takeaway of this is that for small studies, categorical outcomes are not very powerful. For larger studies, it's less of an issue. But if you're trying to show a statistically significant improvement in a small study and you've got categorical outcomes, it's going to have to be a really big difference. Talk a little bit about study protocols, sample size, and power. Generally speaking, every study needs a discussion, at least a discussion about sample size justification. So if it's a pilot study, you might just be saying we're just going to have 10 per group, but you still have to discuss that at some level. Most will require sample size calculations, I would say, and most need you can demonstrate sufficient power for your aims to any agency, whether it's the IRB, the FDA, NIH. Most investigators often like to have statisticians assist them with this. Some of them really don't know how to do power and sample size. It's just not something they do on an everyday basis like statisticians do. So that's something that I get involved with lots of people. When I first get introduced to investigators, they're looking for study design help and sample size assistance. So again, generally speaking, most studies need to demonstrate that each specific aim is sufficiently powered, not just that the first one is. And so you need to think about that. You need to typically demonstrate power for all of your aims. This is a pretty common issue. So just sometimes, this is just one example. If you've got 3 aims, one of the strategies is to designate one of them as a primary outcome. you probably have one thing that you really want, the very first thing you want to show at the end of your study at a conference. What is that? That's probably your primary outcome. And if you do a power analysis on that, and you say that that has sufficient power, then for the other ones, you can just show, given conditionally that you have powered for your primary outcome, what can you do for the other ones? So you would present minimally detectable differences for those other aims. And we do this all the time with NIH grants. The other thing people like to talk about are, doing subgroup analyses. This is a funny category. Doctors call them subgroup analyses. Epidemiologists call them effect modification, and statisticians like to call them interactions. But they're all the same thing. That they're basically trying to say that the treatment effect is better in one group than another. It could be maybe the drug works better in younger people than older people. It could be, that it works in, just for some reason in different groups better than others. Typically, this requires almost a doubling of your sample size if you want to be able to show statistically that these effects are different. So you got to be really careful with interactions and subgroup analysis. One obvious example is a lot of pressure from, let's say, NIH to look at gender effects. A lot of people say, we will look at gender effects, but the study is not powered for gender effects because it would need to almost double in size. And of course, that's often impossible to do. Here's an example of where we got data. This was for a recent R03 that was done here in our Convergence Institute. We didn't have any data for WOMAC scores, and those are like an overall score for like arthritis and pain. So we found this article, and so that is our reference article, because I needed this number down here. I needed the standard deviation in order to do power size calculations. So it was identified from one of the co-investigators that we could use this paper. I told them we need to reference where we're getting this information. So this was the paper. You can see that number 13 is going to carry over to the next page. Over here, we show that this is actual text that was in the grant application. So we referenced the paper. Here's the Lee reference, and we said that in that study, the standard deviations were about 13. Then we have this table down here. We show the reference again. Here's that 55% difference. Here's the standard deviation of 13 again. And that 55% difference relates to a delta of about 20. We powered on that, and here's the power, 96% power. So you don't always have to have preliminary data, but you have to have some data from the literature that is similar to what you're going to be doing. So that's an example where we took data from the literature for an NIH study. We'll see how it goes. It's under review. It's good to challenge your own assumptions. What happens if you underestimate your group differences by a little bit? What if you only recruit 75% of your target sample, so you don't have, your sample size is shrunk a little bit? What if people are lost to follow-up? What if you've got inflated variance? You think you've got one rate of variance, but you end up having higher standard deviations. One strategy down here is to inflate standard deviations to a higher number. So like in that last example, maybe using standard deviation to 15 or 16 instead of 13. Because you don't want to underpower your study if you can avoid it. So after you're satisfied with your sample size calculations, the final thing would be to think about statistical analyses and a plan and a strategy for each of your aims and hypotheses. Basically, you're showing reviewers that you have idea of what you're going to do with your data. It's amazing how many people collect data and then they come and see me and they had no idea what they were going to do with that data. So these are some suggested titles. You might say, why is this guy suggesting titles for these sections? Because these are the titles that everybody uses, and every reviewer is going to look for sections that have these titles. So that's just a reason as to why I'm suggesting these titles. Here's an example where I focus hypothesis for somebody. It's a clinical investigator on campus. And so this is sort of what my job is to help these people focus, because I have to write a statistical analysis section based on what the variables are collecting, but I don't always know exactly what they're collecting. So here's a depression study, and the primary outcome is this self-perceived stress. And that's typically often in categories of 0 to 4. And then the main predictor variable was going to be depression. And that variable is often indicated, a threshold of 17 indicates you may need treatment for depression. So in my mind, I'm thinking, okay, this is what the data is going to look like. We're going to have a table, and it's going to be, how the people that were depressed or not depressed versus those 4 categories, or 5 categories here of self-perceived stress. So I said to this investigator, so is this what you sort of have in mind? And then she goes, no. I want to use the long version of the PSS with 10 items, and that scored 0 to 40, and I want to see if depression predicts a higher score on the Cohen's PSS. So okay, so now we're talking about comparing the mean PSS levels between depression categories. So then I have in mind, okay, this is what you want. You want the long version with a numerical outcome. Going to be comparing means here. And she says, no. What I really want to do is see the relationship along a continuum of the 2 scales. So not categories. So what this person really wanted was the Beck depression inventory not categorized at that threshold value of 17, but along a continuum when with PSS 1 to 40. Now we're looking at like basically a correlation or a regression model. So that's part of my job, and the statistician's job is getting down to exactly what the investigator wants to study. So every aim needs a corresponding methodological write-up for planned statistical analyses. Most investigators don't know how to do this, and this is where your statistician will help you to put together a statistical write-up. If you're working with a statistician, they should give you a statistical plan that is essentially ready to cut and paste into your study protocol. I would say if they send something like that to you, resist editing it. If you think it needs to be changed or shortened, or get back to them, because I've had people edit my statistical sections and it didn't always result in something that was positive. Here are some of the things at the end of your study that statisticians can be helpful with. Obviously, interpretation of results, statistical models, proper inference. They'll help prevent overinterpretation of results, and they can help you with manuscript preparation. And that is what all I had to show you today. Hope that was helpful. See how statisticians can assist you with your research.
Okay. Thank you very much, Dr. Grady and Dr. Wu.
You're most welcome.
I particularly want to remark that I have dealt with a lot of statisticians in my life and career, and there are very few that are so helpful as Dr. Grady, because he understands the issues what the expectations are, and he addresses those right on. I almost laughed when you said that some folks have edited your statistical plan and it didn't survive the integrity of what you intended. So yes, do please pay attention to what he said. His remarks on aims and powering the specific aims is right on the spot, on the money. I wanted to ask you about that, and that is, if you are going to be so rigorous as to have a power analysis for each of the aims, doesn't that end up, by the end of the day, reducing the number of aims? Because, I mean, what you may be embarking on some things that are not supportable. So it's almost like a disciplinary measure when you design your studies, if you apply that logic.
Yeah, I mean, you want to try to. That's only just one strategy. Is picking a primary and then powering for the primary and then showing what you can do with the other aims. So the other strategy would be to power for the aim that needs the most sample size, and then you're overpowering maybe some of the other ones. But that would bring in like maybe more resources. So that happens every so often when we're trying to satisfy all the different.
Okay. So trying to see here if we have any questions on the chat. I don't see any right now. So and then Dr. Wu, in terms of your remarks with regards to the design of the phase 1, phase 2, phase 3, I think most of the industry is probably working on phase 2, phase 1. Not that many companies in Army are at the point of phase 3, and there has been some guidance from the FDA about how to conduct that. So I was wondering, therefore, what are the reasonable numbers for phase 2, sort of take-home messages that people in the industry should be thinking about when they are, launching a therapeutic product and they just want to do a safety study, it passes, and they're ready to embark on a phase 2. I mean, what are the ballpark numbers that would be expected for that?
Usually it's about 30, 40, depends how many arms you have. And arms is the intervention arms, including, control, placebo, or other groups, or other compatible groups. And sometimes can be up to 80. Still, phase 2 is still fairly small study.
Okay.
Yeah, I would agree with that.
Okay, so that's excellent, because I think that's what we have to keep in mind as we develop this portfolio of products in the regenerative medicine space.
Actually, what we had in that RO3 that we put in, or the R21, it was 20 treated subjects and 10 controls. So we had an unbalanced, purposely unbalanced study. And the idea being, let's learn more about the ones with treatment, put less resources into a placebo group. So we had a purposely unbalanced design.
Mm-hmm. Okay.
And that would be considered, I think, a phase 2 study, and that was like 30 total subjects.
Yeah. Okay. So another interesting angle, Dr. Grady, you mentioned this idea of the interactive analysis of interactions, and that more often than not, the power is not there for the interactions because the studies were not designed for that. But nevertheless, it becomes an interesting sub-analysis that you called exploratory. So what does that mean? It means that the original study, or let's say, did not meet the outcomes, or the outcomes were not met, and now you're turning into some exploratory analysis. So that basically would be research, going back to research, right? Or it would de-escalate the study from a clinical trial type project to more of an exploratory study.
Yeah, that would typically be in more along the lines of an NIH study, where they know they don't have enough power to look in subgroups, but yet they might think there's a difference in some subgroups, and they're afraid to almost be criticized by their colleagues for not looking in those groups, but yet they can't power the study to look in those groups, because they would literally, usually it doubles the size of the study. So they sometimes, right after they talk about the specific aim, they'll say exploratory analysis will include, and then you don't have the power for those. They're just exploratory. They're sort of almost not even obligatory at the end, but it's conveying to colleagues that you didn't forget about these potential differences, but at the same time, the study's not designed to detect those differences.
I see.
It's not, a lot of people do this.
Yes, I think from my own experience, that's usually what happens. And the way I have handled that is, in terms of publications, I have separated the primary study with whatever was found. The outcomes were met or they were not met. Whatever it was, and then in a secondary publication, explore sub-analysis with the caveat that it was not part of the original design, and therefore it's very, if you will, very research exploratory. But I think it's important for the industry to understand that, because there is value in those exploratory measurements. So if it turns out that a formal clinical trial, quote, failed, unquote, because it didn't meet the expectation of significance, there still is knowledge and insight that can come from the sub-analysis, and that can be used for the next study, where you can perhaps design with more control for confounder effects. So these are the resources that we have at the University of Connecticut that we want to share with the Army as part of our cost share. The institution provides support, potential support, consulting, resources for laboratory. We had some of that last week with Dr. Broke, and this week we have the statistical analysis and design analysis. So these are the resources that the members of Army could avail themselves to. So I want to thank again Dr. Wu and Dr. Grady for not only an inspiring talk, but also a very practical, down-to-earth reality check on how we should be conducting these clinical trials. I invite you to come again next week at the same time. That will be at 12 noon Eastern for the last seminar on the series, which is one I will be doing myself, and that will be on precision medicine approaches to regenerative medicine clinical trials. So with that, thank you, Army membership, all the attendants, and our speakers. See you next week, Tuesday at 12 noon Eastern. Thank you.